Hello, hello. These are my answers for the essay questions for Unit 1. So our first question is define homeostasis and give an example of how it might work in the body. Homeostasis is essentially, don't mind me as I'm reading off my notes here, the ability to maintain stable internal conditions, like keeping everything running and tight while things are happening outside of the body. Say on a hot day, your body will want to keep you cool. On a cold day, your body will want to keep you warm. So that is homeostasis. And that goes as well for the cells. The cells in our bodies are exposed to different conditions outside of the cell on the external cellular liquid. And so our cells have to be able to keep intact and keep functioning as they would even though things are different inside or outside of the cells. So that opens up the next point, which is the reflex control pathway. And this pathway is our body responding to different stimuli that we receive. And so part of that is we will have a receptor, and that will receive stimuli. And that stimuli is then sent in a signal to what's called a control center. And that control center then gives a signal to an effector to change something within the cell. And that creates what is called a feedback loop. So into our next point, negative and positive feedback loops. These loops are different, but they seek to change something within our bodies based on external input or external stimuli. So our first one is a positive feedback loop, and this is where our body or our cells are trying to move away from a set point. The first example that came to my mind when I started these was a fever. When we have a fever, our body is looking to get away from its normal regulated body temperature to a much hotter body temperature in order to kill off any bacterial or viral infectors inside of our bodies. This is a move away from our set point of body temperature to a much higher one. Another good example would be blood pressure. This is one that we discussed in class. When there's a loss of blood, our body will seek to bring that back to normal blood pressure because without as much blood volume, we drop our pressure. With that drop in pressure, our body senses that and wants to bring us back. And so in a positive feedback loop, it would be seeking to bring us back, but it is actually starving the heart of nutrients, causing our set point to be moved away from farther and farther. So our body actually dies off. Our heart is starved of nutrients that it needs in order to continue functioning. That is a positive feedback loop. A negative feedback loop is when we are moving back to set point. Just like in a fever, I go back to that example, it's a good one. We are going from our hot body temperature back to our regulated normal body temperature of about 98 degrees Fahrenheit. Again, with the blood loss example, if we lose blood and everything else besides our blood loss is okay in the body, then we will see the body constrict the blood vessels in order to up the blood pressure. Our heart will start beating faster to pump more blood, and that will make up for that loss in blood volume. So that is a negative and positive feedback loop. Positive, move away from set point. Negative, move back to our set point. All right, so in part 2 of this essay, I also need to describe our 3 types of protein-mediated transport. And this is something that we went over pretty extensively in class. Forgive me, I'm just going through my notes here. So in our 3 types of transport, we have what's called diffusion via channels. That's one. Carrier mediated via carrier proteins. And our last one is active transport. So our first one, diffusion via channels. This is when we have several channels within our cells that allow things in and out. Diffusion essentially is when we move from a high concentration of a solute to a mediated middle ground of solutes, or what's called, forgive me, I forget the word, our middle point of solutes inside and outside of the cell. It moves through channels or pores that are located in our cells, and this is what gives us our our diffusion via channels. Fish tank. Our next one is carrier mediated, and in this one we have things that need to move in or out of the cells, and so our cells will provide us with a way for things to move in and out of them via carrier proteins, and these carrier proteins will have what needs to be moved in or out of the cell latched onto them, which makes them more effective at moving through the membrane of our cells. One good example of this is insulin. It needs to be attached to a certain protein called GLUT2. GLUT4. And when GLUT4 is attached to the insulin, it moves in and out of the body, dispensing that insulin as the cell needs. When proteins are laden with lots of things that they need to move in and out, they are called saturated. They're called saturated carrier proteins. And this is how they move in and out of the body. And that is our carrier-mediated transport. And our last type of transport is active transport. And this one is done using ATP. ATP is our energy for our cells that we need. Our ATP essentially opens a pump. And this pump, the example that was used, will move sodium and potassium in and out of the cell. It'll move sodium into the cell and potassium out of the cell. And so when these things are moving in and out of the cell, they need energy to do it because they are moving against the gradient that has already been established for them. And so that energy is required to move them in. I imagine it's like packing a suitcase, where the suitcase already has too many clothes, and you need to shove some more in. It takes some energy to shove those clothes into the suitcase. Or like taking out something stubborn that's been stuck in a jar for some reason. Think about getting your hand stuck in a jar, and you can't get it out very easily. You need to pull on it. That is active transport inside of your cell. Just an example where you need to exert energy to get that hand out of the jar. So those are our different types of transport. There's also secondary active transport, and it moves things against their concentration gradients just like primary active transport does. But ATP is not directly involved. Instead, it uses energy that's already stored inside of the gradients to move the solute. So I'm trying to think of a good example. I can't at the moment. All right, going back to my notes. Sodium always has a higher concentration outside of the cell, and so that gradient is used to move sodium into the cell. Because it's so concentrated outside, it will force it into the cell. All right, so our next bit on this question is the concepts of specificity, rates, energy sources, competition, saturation, direction. Looking at specificity, just like with our protein-mediated or our carrier, my mouth is dry. TA, whoever you are, sorry, my mouth is dry. Whatever our carrier-mediated protein is, it needs to be specific to what type of channel it is moving through. It can't just be any protein into any area into the cell. It needs to be a very specific type of protein. Just like I said, GLUT4 is what moves insulin into the cell. That is a specific one. It can't be anything else. It needs to be that specific protein. Looking at rates with our passive diffusion rates or our channel rates, that is going to be a lot slower than our active transport. And then looking at active transport specifically, primary active transport is going to be faster than secondary active transport. The rates are different there because of the energy that's involved. And so because no energy is being exerted into our channels, into our channel diffusion, because no energy is there, it's taking a lot longer to move through those channels into the cell. Energy sources, ATP, is used as well as the gradient that I mentioned. If there's a higher concentration outside of the cell of sodium, it wants to move in because of that higher concentration, and so it will. If there's a lot of potassium inside of the cell, it will want to move out because there's too much. It wants to move to where it's equalized at that point of equilibrium, the word I couldn't remember just earlier. Competition, sometimes different proteins will want to enter the body or the cell at the same time, and so they will compete based on saturation of which can enter the cell. And then saturation, just like I said, when a protein becomes laden with different things that is carrying into the cell, it is saturated. That is the answer for question 1. I will be back here with question 2 in just a moment.